Okay, so I love this cartoon. It's one of my favorites. Take a second to read it, and because what it really does is frame what we're going to talk about, right? Because it's like sleep is clean air and it's clean water, and you never think about it ever until you don't get it, and then that's all you can think about. Oh my God, why can't I go to sleep? So you guys, you don't need to read all of this, but basically this just is a little synopsis of the last 5 years of pretty good research about what's the prevalence of sleep disturbance in ASD. This says children, but it could just say people. And the bottom line is that every time you look, whether it's 500 subjects, it could be 5,000 subjects, the data is pretty clear. The incidence, the prevalence of sleep problems in ASD is just higher than the typical population. And so what are those problems? Well, they could really be anything. They run the gamut. Some of them are sleep-disordered breathing, but by and large, we see two major things: problems with insomnia, which is actually a bigger category, and problems with restricted sleep time or decreased total sleep time. So the insomnia problems, right, difficulty getting to sleep, so these really long latencies, or maintenance problems, sleep maintenance problems where you're up multiple times during the night, or these terminal insomnias where you wake up at 4, you're up for the rest of the day, and there's no naps involved. And these are obviously huge problems, and there's some great research going on addressing how do we make this better for people. Beth Mallow and her group at Vanderbilt, the Autism Treatment Network, there are great researchers looking at this, trying to ameliorate it from sort of a clinical point of view, right? A research, not a research, but a clinical sort of treatment point of view. And that's actually not what we're going to spend the next 10 minutes doing. The next 10 minutes, I just want you to think with me about something totally different and reframe this question. So why won't my child sleep? It's actually, unfortunately, a really easy answer. And the answer is, we don't know, right? We don't know. That's why we do this research. But I want you to think with me about the way I look at sleep, which is, what is it about the way my child is not sleeping that's going to inform me about the way their brain is developing? Because I think that's a really rich area, and we need to think a little bit more about that. Because if we think more about that, then we may be able to get back to that question. So this is a photograph that I love. This is a very old photograph. On the left is my niece, who was 7 in that, is it the left? Yeah, who was 7, now she's 26. And on the right is my daughter, who's 19, and she was a month. And I want you to look at that and see what I see, which is little girls working really, really hard, right? They are working so hard. They have no idea, is the van moving? Is it stopped? Is mom taking that picture or not? They have no idea because they're doing the activity of childhood, which is sleeping, right? So 16 to 17 hours out of every 24-hour period in infancy is spent asleep. The average 2-year-old will have spent 9,500 hours, that's 13 months asleep versus 8,000 hours awake. And between 2 and 5, the time that you spend asleep equals the time that you're awake. You see where this is going, right? And then on from there, from like 5 to 16, 40% of your time is sleeping. So the activity of childhood is sleeping. And sleep is the primary thing that your brain does during early development. So as a neurologist and somebody who's studying brain development, I thought I was going to be able to open up some compendiums and find some libraries and figure out some more information. But we just don't have a lot of info about that early period and about what sleep is doing. So these are some great Golgi plates, and what they show here is just, this is a child, these are pyramidal neurons, right? So this is a child at a month, a child at 4 years, about the same age of those girls that I showed you in the photograph. And this is just to show you that there's an enormous amount of postnatal growth in cortical connectivity. Your brain is continuing to build and grow during this period, and we just don't know enough about the role that sleep does. We know it's important, but we need to know more. And so at the same time that we're strengthening these connections and the foundation sort of anatomy of the brain is growing, there's this rapid shift in sleep architecture, a major reorganization. But you can see from this little cartoon on the left here that between 0 and 5 is when most of that is occurring. And I don't know if you can see it, but there's a little white hash mark there, and that means that after 5, those sleep states pretty much are the same. From 5 until the rest of your life, the amount of time that you spend in REM, non-REM sleep, or awake is pretty much stable. And so to me, that says there's this massive reorganization of the brain that's occurring between 0 and 5 that we don't really know enough about. And the important part about that is that it's not just whether you're dreaming or not dreaming, it's that REM sleep and non-REM sleep, they have different neurotransmission. They have different protein translation. There are different genes that are being expressed during that time period, different hormones. There's a ton of information that we need to know more about. and different membrane potentials, because the language of the brain is actually electrical. Here's another pyramidal neuron. This is how the brain talks to itself, right? So we can measure the changes in membrane potentials per second in an area, and we call that, when we measure it from the scalp, the EEG. And this is a little girl in our lab, and we've got about 1,000 now of these with little kids. And when we measure at the scalp, the EEG, we're getting the postsynaptic activity for hundreds of millions of billion neurons. And that's a ton of information. So I just want to play this in the background when I tell you a little bit of a story about how I came to be interested in this. So this is what I learned how to read on. And you can see in the bottom, these are 30-second epochs of sleep. And when I started at the NIH in 2006, I was really interested in being able to read my own data. And these little brain waves, you have to figure out with what are the different signals, what are the different oscillations, what do they mean? It's really an exercise in decoding, right? And so you figure out how to integrate all the rest of the neurophysiology with the rest of the body, the chest, the legs, the heart, and you come up with a little hypnogram that tells you, this is how this person is sleeping. So I had this wonderful opportunity when I started to train under the then chief of the EEG lab at NINDS, a guy named Dr. Susumu Sato, and he had a particular interest in neurodevelopment, but also in sleep. And we sat up there and we read hundreds and hundreds and hundreds of these. And he taught me how to stage and how to figure out what was actually going on. But at the same time, the data that I was reading happened to be this incredible gold mine of information that had been really, really deeply phenotyped by a colleague and friend of mine, Dr. Audrey Thurm. I know many of you at the table know her. And she had this group of kids who had autism, kids who were matched for nonverbal IQ, who had developmental disability but did not have autism, and typically developing kids. Their all average age was about 4. And so I was learning on that cohort. And so when I got pretty good at being able to stage it, I could do, whether it was 4 hours a night that they slept or 8 hours a night that they slept, I could finish a record in about 45 minutes. But then, maybe every 5th record, it took me hours and hours to stage, like 4 or 5 hours, an entire morning. And I would say, Dr. Sato, I think this kid has autism because the signals don't look right. This is probably one of our autism kids, right? They're so disrupted. We're not doing the normal cycles. There's no REM sleep here. And when I was right, about 4 or 5 times after I was right, we said, ooh, maybe we ought to do something about this. And we actually sent the whole cohort up to NYU to be read by another colleague of ours who actually works with Dr. Davinsky, Dr. Alcibiades Rodriguez, and that was our first paper. And so we published that, and we were able to actually pull out the kids who had ASD from the other 2 groups, just on the basis of something superficial like how much time they spent in REM sleep, and that was a REM deficiency paper. So that galvanized my interest, and it continues to sustain me, because what I think is there's a ton more information in that nighttime data than we know, and we leave it on the table in most labs. Here's just another example of what we did. This is a little cartoon showing you the thalamus, right? It's this little deep structure deep in the brain. And when you leave sort of light sleep and you go into sort of N2, we call it, or stage 2, which is sleep where you're not really taking in any other environmental cues. It's the gateway for consciousness. You're now asleep. And when that happens, at the scalp, you get this little third line down. You get a really particular signal. It has a particular amplitude. It has a particular number of oscillations. There's a duration. It's called a spindle. And the spindle represents a microcircuit in the brain between the thalamus and the cortex. It's mediated by GABA. And there's been a ton of information saying that the health of that system and spindles in general have to do with learning, memory, and cognition. And it's what a lot of sleep researchers look at. And in the last couple of years, actually, there have been about a dozen papers looking at the health of that particular system and measuring spindles and showing that in schizophrenia, there's a spindle deficit. So we went back and looked at that cohort that I told you about. And this little panel up here, I just want to show you. This is our most recent data, hot off the presses. You can see that deep blue line in the bottom there, how far below it is from the other 2 lines that are basically statistically not distinguishable. That's the spindle density in our little kids who have ASD. So again, we're looking for sleep-dependent signals, and we're finding enormous differences. So we were excited by that. Here's another example of just one of the things that we looked at. So this is kind of a complicated little heat map, but I'm just going to run through it with you, and you'll see how easy it is to read. So we put 19 leads on the head, and we record from those situations. And if you compare each little lead, maybe this one here, and this one, and then that one, you work out the math, it ends up being 171 different electrode pairs from the front of the brain to the back, right? That's the left panel. Along the bottom, sleep states. Awake, REM sleep, slow wave sleep. Warm colors means there's a difference. No color or black means there's no difference at all. So this panel is kids who have ASD, kids who are typically developing, they're 4 years old. When we looked at the awake state, you see just black. That says we don't see any difference in the way that any of those little areas of the brain are oscillating or talking to each other until we go to sleep. And then when we go to sleep, we found hugely marked differences, region-specific, when the brain started oscillating in the slow wave sleep manner. And so we thought that, wow, that's really interesting. What is going on there? And the bottom line with all of this is that changes in sleep electrophysiology in ASD are real, whether it's coherence or spindles or stages, and we're not really sure exactly what they mean, but what I'm really intrigued by is that we don't look. And why aren't we looking? So more and more people are realizing that there's an enormous amount of information in the way the brain is talking to itself and building itself at night, and that approaches to analysis need to catch up. And so we're working with computational modelers to move beyond visual inspection, to move beyond coherence, and actually to use that data, we should be looking for algorithms that we can't see by eye that might inform a little bit better. Because this is really the bottom line, I think. So anatomy is great, and the structure is important, and those Golgi plates and things that you can get from the MRI, that's all really useful. But the beauty of the brain, right, is that it's creating and then engaging and disengaging these dynamic networks that come on and off in response to the environment, and that's actually what we're measuring. So when we talk about cognition or social cues, that's what we're looking at, the ability of the brain to do that. So we think we can use sleep neurophysiology as this more proximal biomarker between genes and disturbance. So you say, why doesn't my child sleep? Well, we're not sure. Between structure and sleep disturbance. Sleep neurophysiology, whether or not it's circadian abnormalities or the information that we're getting in the neural oscillations, or even something simple like stages, so that the next time that I give a talk like this, I can say, well, your child with autism doesn't sleep because they have a clock gene abnormality and it is predisposing them to early morning awakenings and they're only going to sleep 5 hours a night. But your child with autism, actually, they're not sleeping because they have a brainstem abnormality, and it's a non-REM problem that wakes them up in the middle of the night. So the answers are going to be different, and they're going to be very specific. But if we don't fill in this black box, I think, again, we're just leaving information that's really valuable on the table. Because I think what we're all looking for is a way to figure out, before this behavior is manifest, or maybe not before it's manifest, but before it's exacerbated or augmented, how are these networks being malformed? Or how are they not adapting the way that they might? And a way to get there is this project that, again, my colleague and I, Dr. Adrian Thurm, have launched. Last September, we started with the very first inaugural Sleep and Neurodevelopment Consortium. And you can go to that link and you can see all the folks that came and talked, and multiple different disciplines. We had psychiatric geneticists, the engineers, and the computational modeling folks, and there's summaries of all their talks. Because in order to move forward with those answers, we need to actually fill that data gap. We need to figure out between 0 and 5, what does the brain look like when it's building itself during sleep? Because, sadly, we don't have that information yet. So I think the bottom line is that sleep is still a mystery. It's a mystery for adults, but it's a huge, big black box for kids. We just really don't know. It's a behavior for sure, and that's what we see, and that's what we complain about. But it's a process, and the process of sleep is something that you can absolutely objectively measure robustly over and over and over again with the EEG. You can get there. So it's a process. It's also a function, right? And to figure out what the function of sleep is, we need to be asking these questions with a multidisciplinary group, which we're doing. But the last thing I want to leave you with is it's also it's a program. So the brain is wired to run sleep as a program every night on the foundation that it has. And when that program stops or doesn't run correctly, which is what we're seeing in people who have lots of different neurodevelopmental disorders, there's information in the way that that program goes awry. And that's the focus of our research. So thank you for listening. That's it.